Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Managing Multiple Complex Molecules in CDMO Projects. My name is Samaya and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please send in questions using the questions chat box in your control panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank ProBioGen, who developed the content for this presentation. ProBioGen is a leading CDMO and a specialist in developing and manufacturing biopharmaceutical active ingredients, viral vectors, and vaccines, combining both state-of-the-art development services based on ProBioGen's CHO-Right expression and manufacturing platform, together with intelligent product-specific technologies, yield biologics with optimized properties. Rapid and integrated cell line and process development, comprehensive analytical development, and following reliable GMP manufacturing are performed by a highly skilled and experienced team based in Berlin. Now it's my pleasure to introduce our speakers for today's event, and our first speaker is Annika Lubetz. Annika Lubetz is Director of the Bioassay Department at ProBioGen, where she's accounting for the development, optimization, and qualification of various bioactivity assays for potency characterization of biopharmaceuticals, such as engineered monoclonal antibodies or fusion proteins. After graduating as a biotechnology engineer, Annika Lubetz joined ProBioGen in 2005. She worked as a research scientist in the Cell and Tissue Services Department. There, she got involved in numerous projects dealing with immune cell expansion, drug exposure, and analytics before she engaged in the generation and investigation of in vitro immune responses using human 3D models. And our next speaker is Dr. Thomas Rose. Thomas Rose, PhD, is Head of Expression Systems at ProBioGen. He received his MSc in biotechnology from the University of Applied Science in Berlin and his PhD in biochemistry from the Free University of Berlin and joined ProBioGen in 2001. His work focuses on cell line development and expression technologies for protein drugs. He was instrumental in establishing the Freedom CHO-S kit as well as ProBioGen's CHO-Right platform and pathway modulator and directed luck technologies. He also has extensive experience in cell line development for a variety of complex proteins. And our next speaker is Dr. Ellen Hilgenberg. Dr. Ellen Hilgenberg is the Senior Business Development Manager at ProBioGen. She studied biology and received her PhD in immunology from the German Rheumatism Research Center in Berlin, Germany. Afterward, she completed her postdoc at the Max Delburik Center in Berlin before she joined ProBioGen. So without further ado, we'll go ahead, attendees, and start the poll questions. So we have our first poll question for you on your screen now. So you should be seeing that poll question. And that poll question is, who do we have in the plenum today? And you can select one here. So your options are, one, I work for Big Pharma, or that could be I work for a biotech company. That could be I work for a research organization, or I work for CDMO myself, or something completely different. So go ahead and click on the option that suits you best. So once again, that question is, who do we have in the plenum today? And you can select one here, and you can do that just by clicking on the option that suits you best. Okay, so I'll give you a couple more seconds, audience members, to answer this poll question, and then we'll go ahead and see the results. So I see the majority have voted, so I'll go ahead and close the poll and share the results now. So the results are as follows. 13% of audience members voted for the first option, that's I work for Big Pharma. 33% voted for I work for a biotech company. 20% voted for I work for a research organization, 13% also voted for I work for a CDMO myself, and then 20% voted for something completely different. So thank you everyone for participating. And then we have our second poll question for you. And that poll question is on your screens now. So that question is, how much experience do you have with this kind of projects? So you can select one here. So that's none, or I was involved from a distance, or I dealt with it very few times, or that could be I'd call myself an expert. So once again, that question is, how much experience do you have with these kind of projects? So you can select just one here, audience members, and your options here are none. It could be I was involved from a distance, or I dealt with it very few times, or I'd call myself an expert. And just do that just by clicking on the option that suits you best. So audience members, I'll give you a couple more seconds to answer this poll question, then we'll go ahead and see the results here. Okay, so I see that most of you have voted, so I'll go ahead and close the poll, and then we'll go ahead and see the results now. So the results are as follows. 29% of audience members voted for the first option, that's none, and then 43% voted for I was involved from a distance. 21% voted for I dealt with it very few times, and then 7% voted for I call myself an expert. So thank you everyone for participating. Now we can go ahead and start the presentation. So speakers, whenever you're ready, you can go ahead and get started.
Okay, then thank you so much, Sumaya, for your nice introduction. Hello to everyone also from our side. A big thank you for joining our webinar today, focused on navigating the intricacies of complex molecules and CDMO projects. We heard it already, nevertheless, it does no harm. I'm here alongside with our two technical experts. We have Annika Lubitz, our bioassay wizard, representing ProBioGen CMC unit, and we have Thomas Rosa, our specialist in cell line development and a transposase enthusiast. So today's session is designed as a panel discussion between our experts and me. And throughout our webinar, you can expect in-depth insights, practical guidance, and real-world experiences shared by our esteemed guests who have actually successfully managed such complex molecule projects. So it doesn't matter whether you are an industry professional or whether you are an aspiring newcomer. I think there's something to learn here really for everyone. And with this, I would also like to start with a short introduction, and I will go back in history a little bit. As you might know, it all started in 1986 with Muromonab, an anti-CD3 antibody that actually paved the way as the first commercially approved monoclonal antibody. And since then, the world of antibody-based drugs has evolved rapidly, and it became a crucial player in the pharmaceutical scene. Now, approximately a decade ago, multi-specific antibodies started to gain traction among drug developers. And why was it so? I think because mainly they can tackle multiple protein targets at once. They are offering the promise of more effective and safer treatments. And other exciting results are improved specificity and targeting across a spectrum of diseases. But with this expanded capability comes also greater structural complexity. So we have more charge and structural variants and a potential array of post-translational modifications. Drug designers now indeed face the challenge of finding optimal binding structures, the right half-life for specific biological needs, and also addressing steric consideration for engaging multiple targets. So it's indeed really a puzzle, given the multitude of variants linked to these complex structures. Therefore, our goal today is, and I know I said it before, but the goal today is to provide you with insights into navigating the production of these intricate molecules. What workflows and solutions are necessary to swiftly and efficiently bring these innovative molecules to the clinic with top-notch quality. So from where I stand, it's not just about the rising request for complex molecules. There has also been a growing trend of simultaneously launching multiple molecule variants. And I know that such requests have been around in the past already, but they are more frequent and often coupled with highly complex molecules even. It's an exciting but also challenging landscape we are exploring together today. you get ready to elevate your understanding, refine your strategies, and also unlock the potential and success in your own projects. And the panel discussion is now open, and I would like to address my first question to Thomas. Thomas, perhaps you can enlighten us why the demand for multi-parallel development is increasing so much nowadays.
Thanks so much for your introduction. And it's also great to have so many people here in the audience. Like I know for some of you, it's quite early, and I really appreciate you taking time. And time is also a good password to get started, right? So Alan, to answer your question, I think the main driver is time. So smaller companies often have ambitious timelines to reach their funding milestones. However, by the time you enter clinical development, you may not have acquired enough data to decide on a clear lead variant. But of course, in a competitive world like ours, time is always pressing, like for everyone. And don't get me wrong, but I think that this is all the good thing. I mean, it's about developing efficient drugs and getting them to the patients as quickly as possible, right?
Absolutely. Time is definitely crucial. But how do we effectively save time now?
Good question, Alan. And the answer is actually quite simple. You can start your projects earlier. Perhaps your latest animal study is still running for a few weeks. If you start with multiple variants, you can finish your study and still make decisions based on the outcome. And there is another important aspect. Should one variant fail, you don't have to repeat cell line development from the scratch. I've also seen manufacturability issues of individual variants that were not apparent at earlier development stages. I know it sounds unlikely, but it happens. So the point is that we are not just talking about faster time to clinic, but also about risk mitigation. And I think time and de-risking are great reasons to kick off with multiple variants and select the final lead later.
Thomas, let me just touch on the topic of de-risking. I think it's important. So what I've heard so far is that de-risking should be incorporated as early as possible. And as I understand it now, one option is to start with multiple complex molecules in parallel as well. Are there any other differences in extra challenges in such a complex project compared to a standard molecule? And if so, can you please explain?
Well, I would say it's the nature of these projects what makes them different. Like think of a bispecific 3-chain antibody and a start with 4 variants. First, you have to subclone multiple genes into expression vectors. Perhaps you also have to test multiple chain ratios to achieve the highest heterodimer content. Then, analytics adds another layer of complexity and may even require a unique setup for each variant. So to cut it short, these projects are less predictable and you need a lot more resources.
Okay, understood. So considering this unique aspect, let's dive into what this implies for CDMO projects dealing with multiple complex molecules. Thomas, Annika, you know we have discussed already before how to best approach projects with multiple complex molecules, and you identified actually several key takeaways, 5 to be precise. And the first one is building solid partnerships. The second one is tapping into scientific expertise and experience. Third would be leveraging top-notch platform processes and stellar technologies. Fourth, ensuring seamless integrated in-house processes. And number 5 is having a robust in-house analytical team. So now let's explore these crucial credentials a bit more. Annika, let's start with you. Could you shed some light on what exactly is involved in building strong partnerships?
Thank you. From my point of view, partnership is a very good term because that's what it's all about. It means close interaction with the customer, and this can best be done with experienced project managers. They should have strong management skills on the one hand and technical expertise on the other hand to be able to answer questions directly, for example, instead of going back to SMEs for each and every issue. In addition, the customer brings in its own special expertise, but sometimes you have to ask the right questions to fully utilize it and push the project. Another interesting and useful tool is the person and plant concept at certain stages. If this happens during production phase, it avoids delays in time-critical decisions, which could be challenging in case the customer is located in a different time zone, for example. And if during production, for example, the cell viability in the bioreactor is still very high and the cells are still productive, the process could be prolonged a bit, leading to an increased overall yield. But this must, of course, be approved by the customer. And finally, such visits promote even deeper customer interaction and exchange.
And what other steps can enhance understanding customer expectations, Annika?
A lot more can be done, but most importantly, it is quite crucial to understand the client's expectations at the very beginning of the project and to manage those demands in an appropriate way. First and foremost, this includes timelines, and of course timelines, but in addition to that, quality and quantity requirements must also be considered. And to address all these things early on, a reliable proposal covering these demands should be set up.
Annika, I totally agree, and would just like to add that it's also very important to understand the molecule. Clients have already produced and analyzed transient material before we start a project. So there is early data, and that should be considered to address potential challenges and critical quality attributes right from the start. And this is extremely helpful to streamline and facilitate further development steps.
So what I've learned is that having experienced project managers is a real game changer. And it is absolutely critical to understand not only the customer's expectations, but also the nitty-gritty details of their molecule right from the start. Is there anything else that makes a great partnership, Thomas?
Well, for me, true partnership also means flexibility and willingness to go the extra mile. You know, sometimes you simply have to shift the focus driven by results. And if you face unforeseen challenges, you need to deploy additional resources, sometimes even beyond the contracted scope. And I can tell you that surprises are not uncommon in these projects, which brings me to my last point. A good backup plan is also very helpful to contain potential risks. I recall even cases where it saved the project.
Thanks to both of you. I would now like to close the chapter on building strong partnerships and move on to the second key credential we identified before, the scientific expertise. So scientific excellence and expertise are said to be particularly important, but how do you see that, Annika?
I think there are a few general aspects. First, a sound experience with this type of projects is of essence. It's not like you foresee every possible challenge from your experience with previous projects. However, if you know your platform and the structure of these projects, you also know the critical steps and potential pitfalls. And then you will be able to anticipate measures and adjustments that may be required. Also, with every new project, you gain knowledge and optimize your processes.
Thomas, have you got any additional insight or thoughts to share?
Like I said, you really have to look at the molecule and understand its structure and format. The clients are the experts on the molecule and of course the data that was obtained during drug discovery. So get the teams together and discuss the data that is available, because then you can anticipate critical product features and establish analytics and strategies to control them.
Does a deeper understanding of the molecule format also play a role in CMC?
In any case, it helps to know critical scalability steps. Doing so, you can maintain process performance and also product quality over various scales. Also, it is really recommended to identify test parameters that are different at larger scale and that can already be tested in the small scale devices. So the selection of an appropriate downscale model is definitely a key element.
Thanks, Annika. Thomas, the last words in this section belong to you. Are there any other skills that are useful that you would like to mention now?
Well, I think it's also very important to have strong problem-solving skills. Let me give you a quick example. We received a cell line that was generated somewhere else. The product had a really unusual and unwanted post-translational modification. When we looked at the problem, we specifically modified our own process media formulation and developed a completely new downstream strategy. This combination was pretty successful, and we were able to eliminate the unwanted species.
Great. So let me sum up what you two just said so that we are on the same page. We learned about the necessity of experience. The more experience one has, obviously, with different molecules, the better one can react to new challenges. Additionally, it involves truly delving into the format and its structure, which brings benefits both in the cell line development and in manufacturing. And last but not least, the right attitude obviously is essential, finding pleasure in the challenge and in solving complex questions. So let's move on now to the third credential, platform processes and technologies. And you said that managing multiple molecule variants represents challenges. So tell me, what's needed in platforms and processes to handle them effectively? Thomas, maybe you would like to start now.
Sure. Great question, Aaron. And there is a simple answer. Capacity for parallel handling is essential. That may sound trivial. However, it's not just about high throughput. What you also really need is streamlined and integrated approaches paired with scientific expertise. And this applies across all process steps, from efficient vector cloning strategies to automated cell culture, as well as high throughput purification and analytics.
So robust yet adaptable platform processes are crucial. How does this now apply to CLD?
You know, in cell line development, the platform provides a solid foundation. However, being able to make instant adjustments is equally important. But let me illustrate what I mean with flexibility with an example. We have automated the workflow of single-cell cloning and escalated clone screening. The system does what it was built for, it decreases the throughput, saves a lot of time, and also delivers better clones. However, here my point is that this is really an open system. That means we can flexibly adjust or exchange processes, methods, and even hardware if required. So in the end, in cell line development, nothing is really fixed except quality.
Does manufacturing differ, Annika?
I totally agree with Thomas first. Platform processes also exist for upstream and downstream processes, initially based on standard IgG molecules. However, complex molecules often require adaptations or partial application of these steps. To be able to control this as early as possible, it is necessary to identify the critical steps, raw material, and measures. Doing so, you don't waste time on platform steps that are already working, but spend the resources on the additional steps that really matter.
Can you illustrate this maybe with an example, what you just said?
Yes, of course. Just think of the upstream process. Sometimes it comes to fragmented variants upon loss of cell viability. Therefore, keeping the cell viability high sometimes becomes more crucial than usual. Then you also have to keep a close eye on the time of harvest. Aside from that, a sound control of focusylation level during upstream processing must be ensured, since it could have a direct impact on the activity of the antibody, at least and substantially in ADCC activity, which is known to be the most affected by the Foucault's level. To give an example for downstream processing and virus inactivation, it is essential to adapt the protein A elution conditions or use alternative treatments depending on the sensitivity of the molecules. And finally, flexibility is just as important when it comes to testing and incorporating the latest innovative product developments of suppliers for raw materials, for example, new resins or filter materials, which in turn allows you to stay up to date and get the best out of the molecule. Nevertheless, one premise applies to all steps. Don't artificially inflate the process, but reduce complexity wherever possible.
Great, thanks to both of you. So what I have taken from the last few minutes is that we need a streamlined and integrated process, but we also need to remain flexible. I believe this concept of flexibility is especially important and should be implemented as good as possible. Now I would also like to talk about technologies. Thomas, this is your playground, obviously. Tell us, what's the value of technologies in complex molecules?
Thanks for the question. I think access to the right technologies is key. Take, for example, our directed lug transposase. Because it boosts productivity, the focus of clone screening can now be entirely shifted on product quality.
Sounds fantastic, Thomas. Can you tell us now more about ProBioGen's transposase technology, please?
Sure. We call it also epigenetic targeting. This unique transposase was modified with a chromatin reader domain that binds to a specific histone modification. As a result, it directs transposons to highly active genomic integration sites. As expression is also strictly copy number dependent, there's even a direct impact on bispecific formats. Ratios of transposons used for individual chains can be flexibly adjusted to modulate the product purity. And this is the reason why directed lug delivers bulk pools and clones with highest heterodimer rates. What our licensees also really appreciate, that it's pretty much plug and play. But there is another great advantage. Directed lug also provides highly stable clones. And this is why we took expression stability studies from the critical paths to select the clone for master cell banking. And as you can imagine, that has a pretty significant impact on timelines.
Yes, I'm still impressed by the transposase technology and probably one of the biggest fans of the transposase technology. But are there maybe any other remarkable technologies that you would like to mention here?
Actually, our Climax technology provides another great example. We have been asked to replace a cell line for late-stage clinical development. The challenge was that the antibody was only partially afucosylated. And here we took full advantage of our Climax technology. In contrast to approaches that knock out the fucosylation pathway, Climax efficiently deflects it. So we were able to generate a highly productive clone that produces a fully afucosylated antibody. Because the pathway still works in these cells, the desired level of fucosylation can be adjusted simply by adding fucose in the process. And this is what I mean by saying that you need not just any technology, but the right technology to be successful.
Thanks, Thomas. I really think these technologies alone would probably fill an entire webinar. Let's see, maybe we'll be able to do so soon. Now we have to move on with the program and get back to our credentials. Integrated in-house services were named. Annika, can you maybe point out why this is crucial and how does it impact quality and timelines?
Yeah, like both of us mentioned before, it needs some kind of flexibility to react to client needs that are coming up during the project. For example, some extra material needs, additional testings, but also stability exercises and so on. For that, it is also of advantage for the contractor to have as many project steps under own control. That's the reason why we at ProBioGen integrated more and more own capabilities like buffer production capacities, but also internal potency assay development, which was implemented quite a while ago. We've already set up a wide range of bioassays or can even develop new ones from the scratch, which can be performed in addition to the potency assay, which serves as a release assay later on. And those additional bioactivity results can help to better characterize the molecule. Recently, we increased also capabilities for stability assessment and formulation development. And also important to mention is the analysis of virus removal potential of particular downstream steps. And all these things are of great value, especially in case that we deviate from the platform process for standard antibody. And secondly, reliable partnerships with third parties, such as suppliers or service providers, matter, especially during the times of the COVID pandemic. Supply chain management became just as important as science. It showed us the less you are dependent on third parties and the more you have under your direct control, the better.
Can you point out how this now applies to the interaction of the different service lines?
Well, an integrated approach between cell line and process development, as well as material production and assay qualification, is not only recommended to shorten timelines, but also supports deep interaction of the various teams. And the final clone that is selected should not only be tested for productivity and product quality, but also for its behavior in a controlled bioreactor and for its specific bioactivity, just to give you some examples.
Thank you, Annika. From the integrated in-house services, I'd like to move on to discussing the establishment of in-house analytics, which is also the final item on today's agenda. Thomas, maybe you would like to start.
Of course. I think it's no secret that this type of project provides analytical challenges. There's simply no general approach, and methods have to be developed or adjusted at each stage of the project. But let's just look at very early project phase. Certainly, you have transient material that is highly purified. But if you deal with bispecifics, you also need the homodimers to set up methods. So you make homodimer pools as well. However, you only get them together with the pools which you have to analyze to make quick decisions. That means you only have a few days to develop tailored methods and analyze your samples. So you really need a highly skilled analytics team and of course state-of-the-art equipment.
Yes, Thomas, that's totally right. If we now talk about an example, the production of bispecifics, all of us can imagine that the analytics must be specially tailored in the case. Let's only take the bioassay. The perfect bioassay should reflect the two specificities of a molecule. But these kind of dual bioassays may require more establishment steps in advance to find out the right combination of involved reagents. And bispecifics reveal another characteristic, because not only the heterodimer can be formed by the production cell line, but also the two possible homodimers. And that homodimers behave pretty similar to the main variant in the downstream process. So in order to be able to distinguish between these variants early on, it is of advantage here as well to have analytical procedures established in time, which means that this should already be the case during clone selection. So again, the integrated approach helps a lot here, and together with similar equipment, this also enables seamless internal transfer of all the processes and methods.
But wait a minute here, Annika. So I understood that it is important to distinguish between the desired form and the subspecies. But how do you actually do that? What kind of analysis is needed?
Here, suitable analytical procedures can be RP or HPLC when there are differences in hydrophobicity, for example. But also SciX, HPLC, and CE-SDS could be of usage as well when the variants differ in surface charge or size. For the peak annotation, mass spectrometry can be useful. And if the variants can already be successfully separated via HPLC methods, this can lead you into the right direction on how to separate them on a preparative scale in production. But beside the mispairing of chains, also other variants can occur. For example, unwanted glycovariants, and also fragmented or aggregated forms. Especially aggregates can interfere in the bioactivity assay. Thus, stability of the molecules of interest can also be a challenge, and therefore needs to be addressed quite early with the tools we already mentioned. Formulation development, for example, or screening of manufacturability and potency in the phase of clone selection or even clone pool generation already.
Thomas, is it also important to have an integrated analytics team, an integrated workflow in cell line development?
That's indeed a very good question, Alan. Actually, our cell line development unit has its own fully integrated analytics team. This keeps waste short, and of course it saves time. However, even more important is that the scientists are in close interaction to draw the right conclusions in time. I wouldn't say that everything should be done in-house. However, we have even decided to establish our own next generation sequencing group for extended genetic characterization. And our clients keep telling us that this integrated concept makes a real difference. All this enables us to transfer only clones of highest quality, which I think is another key. In the end, it guarantees project success and saves a lot of time and effort in USP and DSP development.
Thank you so much to both of you. I believe it is about time to close our session now. Thank you everyone for participating in our webinar on navigating the challenges of handling complex molecules in CDMO projects. We really appreciate your time and your interest. Special thanks, of course, to our esteemed guests, Thomas and Annika, for sharing their knowledge with me and also their experience with us. So in closing the session, I would like to recap our key takeaways. Establishing close partnerships and ensuring upfront alignment with the customer is the first essential step. We need the highest level of scientific expertise and experience with respective molecules. And this often goes together with special technologies that can indeed improve product function or quality. A robust and flexible platform process, integrated workflows, and well-coordinated teams form the foundation of success. And last but not least, early investment and strong analytical method development and testing capabilities is crucial. And with this, again, thanks to all of you. I hope you enjoyed our webinar, and I hope you have a lot of questions that we will answer right now.
Well, thank you very much for that insightful presentation. And yes, that does bring us to our Q&A portion of this webinar. So audience members, if you have questions, keep sending them in. I've already received some questions, so I'll start off with the first one. And our first question here is, I understand that automation increases the throughput, but how it also produces better clones? Should I repeat that question?
Hello, my name is Carsten Winkler and I'm the VP for Pharmaceutical Alliance at ProBioGen. And I'm leading the automation project, so I think this question goes to me. Intelligent automation is much more than just transferring manual work steps to a liquid handling robot. The system is designed to measure a variety of parameters that enable the characterization of producer clones from a few cells to a fully automated miniaturized fed batch. And this includes stain-free cell counts, highly sensitive and precise titer measurements, or the measurement of metabolic profiles for several hundred clones. This combination enables to perform AMBER-like processes for 100 or more clones much earlier. And similar to the AMBER, we can take metabolic profiles into account for selection of producer clone candidates. And there's another important aspect. The system enables also product characterization in high throughput mode. To give you an example, we can look at aggregation, charge profiles, or end ligands. That means we can address quality attributes which are specific for the molecule much earlier and analyze hundreds of clones if required. If you would only screen for titer, you may lose high quality clones with reasonable expression but exceptional quality profile. So the system helps us to match and balance several quality criteria and titer for selection. Therefore, the automation provides access to the right data earlier and for a large number of clones. And this is the reason why it delivers better clones.
Thank you, Carsten. We have another interesting question from an audience member, and this question is about bioassays. The question is, how long does it take to establish a bioassay?
Well, that's a fair question. Of course, the establishment time depends on the type of bioassay. If it's a more simple binding assay or a cell-based assay. It depends on the availability of the reagents or proteins. I mean, sometimes you can use commercially available ones, but sometimes recombinant proteins are not suitable or even not available yet. And it also depends on the status of the assay. If there's already a preliminary protocol available from the customer or a platform assay method available at ProBioGen. So that I can roughly say that it can be realized within 2 weeks. It could also take longer, of course, until you have a quite robust lab method protocol, so to say, which might then, of course, be further optimized to be suitable for its intended usage as a release assay, for example.
Great, thank you, Annika, for that interesting question and answer for sure. And another great question from an audience member. That question is about genetic characterization. The question is, can you provide more insights how next-gen sequencing supports cell line development?
Well, I think this question goes to me. So yes, to answer your question, there's two aspects. So first is that you can confirm the sequences of the integrated target genes using NGS techniques like we do it with ONT sequencing. Of course, like even though sequence errors are very unlikely, you cannot completely exclude them. So if you know that everything is fine, it simply makes you sleep better, right? And the second aspect, I have to expand a little further. Like genetic stability is usually analyzed by comparing start and end of expression stability studies. So we do it also and use digital PCR to obtain absolute copy numbers. However, as mentioned in our discussion, we have removed stability studies from the critical path. So the data is not available to support the clone decision. But when we perform like NGS analysis, we also look at structural variations of the recombinant sequences. So let me explain the implication using an example from a project with a bispecific molecule. At the end of cell line development, we had two lead clones that were quite similar. Product, quality, titer, process fit, everything pretty much the same. However, one of the clones had a higher copy number and complexity, which may increase the risk for genetic stability issues, right? And the result was our client dropped this clone. So NGS characterization is also a way to mitigate risks.
Great, thank you, Thomas, for that comprehensive answer. And our next question is about timelines. That question is, you mentioned various measures to shorten timelines. What are your actual project timelines?
I think I will briefly answer that question because I get that quite a lot in business development. Timelines always depend, first of all, of course, on the format and on the molecule we are dealing with. I think it's fair to say that for standard antibody formats, we are now down to, let's say, from gene sequence transfer up to even drug product release, roughly 13 months. This is our fastest timeline. But here also flexibility kind of kicks in. So I think it's indeed important to always pay attention. And for complex molecule, it's not always probably the best way to go with the fastest timelines. But it also depends on what the customer wants and what the main focus of the client or the customer is. So there are some clients that say, okay, we want to go to the clinic as fast as possible. And then we go to the clinic as fast as possible. And some say, okay, no, please pay more attention to develop a really very robust downstream process for our complex molecule, an upstream process. And then, of course, slightly takes longer. But I think it's also It is for me important to mention that we do not make any marketing timelines and that we always stick to the timelines that we agree upon with the clients before we start a project.
Great, thank you, Ellen. And we just got another interesting question, and this question is for you, Thomas. So that question here is from our audience member, and it's from Neha. So thank you so much, Neha, for sending that over. That question here is, and it is, what average titers do you usually obtain? So this one is for you, Thomas.
All right, thanks. Well, titers is of course associated to the molecule you are working with. So what I can tell you is what a platform is capable. You learn when you express like a really good and easy molecule. We did that for like these formats and also some more challenging ones. We obtained pool titers up to 10, 12 gram per liters. Yeah, like in that range for clone pools. But of course, if you work on very complicated molecules, and there are molecules that the cells don't really like, you obtain a pool with maybe 2-3 gram per liter. Others, like the complex bispecific formats we were talking about, like 4, 5, 6 gram per liters. So in that range. So it really depends on the specific molecule.
Thank you, Thomas. That was really interesting. And we just got another great question from an audience member about bioassays. The question is, what kind of additional bioassays are of relevance here?
Oh, a lot of assays might be relevant here. And you really need to take care what kind of and how many assays you select. For example, Fc receptor binding assays can be relevant in the context of mode of action or even serum half-life, but also other assays describing the target binding and functionality of the molecule should be taken into account to make sure that not only Fc-associated functions, but also PAP-associated functions are displayed. And these might also be neutralizing or blocking functions, for example, and all these can also be displayed in a proper bioassay.
Great. Thank you so much, Anika, for your answer. And thank you everyone for sending questions in. And we got tons and tons of questions, but not to worry, if you have more questions, you can keep sending them in. So I'm just going to go ahead and quickly share the contact side here. So go ahead, take down all these emails here. Also take down the QR code here as well. So if you need time, I'm just going to keep this here for now. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. It generally takes about a business day to 3 business days for that recording to be uploaded. It will be sent to you, of course, via email. It will also be on our website, so make sure you are on the lookout for that. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now, please join us in giving a big thank you to our speakers today. That's Anika Lubitz, Dr. Thomas Rose, and Dr. Ellen Hilgenberg for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.
Thanks also from my side. Have a great day.
Thank you, Maya. Thank you for the audience.